Good afternoon, Nishika. How are you doing? So how was the Diwali? Fun filled. You lit crackers. Yeah, due to pollution issues, the cracker lighting was banned. You can say. So today we'll be finishing off the genetics chapter. So at your school, which chapter is going on right now? Like which chapter? Still okay. Good crackers. So have they finished with genetics or they're still going on with it? Today I think I'll be able to finish this chapter. There's only little bit recombination and linkage is remaining. Okay. So after that, which chapter they are planning to commence? Like you had told me that they have taught meiosis in school, cell biology. So we can proceed with that chapter till your school starts next chapter. Okay, so from tomorrow, like from next class onwards, Friday, can I take this meiosis part? That will revise like, in-depth revision of meiosis for you. Till your school keeps this genetics going on. Yeah, so I'll start the meiosis because you have some idea about that. So in next class we'll begin meiosis so that because meiosis is quite related to genetics, like fully related. And since you have some basic idea, you'll be able to comprehend easily also. Okay. So today we'll be doing the last part of genetics. Number one, I'll give you some special tips also. First topic is pleiotropy. These kinds of questions are asked in some SAT and all. So pleiotropy or pleiotropism is defined as, you can say, when our gene influences multiple unrelated phenotypic traits. When one gene influences multiple phenotypic traits. Means there was a theory, if you know, like one gene, one protein hypothesis. But that theory is completely wrong because what happens is we have almost, you can say, 22,000 genes. Okay, almost 22. In many books, you will be seeing that you are having 25,000, but that is a false information. As per the correct thing, correct data, correct current research, we have almost 22,000 genes. But the thing is, we have more than 1 lakh proteins. Still, many proteins are being discovered. Okay, still scientists are unaware of many proteins, and there is a plethora of proteins known as mini proteins. Okay, which are only a few hundred amino acids long. So you have tons of mini proteins which are being discovered, like they are a new class of proteins. So how come from 22,000 genes you are synthesizing more than 1 lakh proteins? So one gene, one hypothesis or one protein hypothesis is completely wrong. What happens is when I'll teach you this mRNA thing, then you will be knowing that in DNA, even in the coding region, suppose this is the gene region of the DNA, still in this gene you will be having this E is referred to as exon, I is referred to as intron. Introns are nothing but intragenic non-coding sequences. Non-coding sequences. So by various permutation combinations that you have studied in your mathematics chapter or your mathematics subject, we can make different RNAs. One way I can only select E1, so one mRNA will be only E1. This will give me protein 1. Another will give me E2 if I only select E2. Okay. This will give me protein number 2. Then I can combine E1 and E2. I will never add I because I will be thrown out of the mRNA, processed mRNA. That is known as splicing event. Okay. So this is known as alternative splicing. I'm splicing alternatively. Okay, E1 and E2 are both are there. This will give me protein 3. So from one gene, you can say 3 proteins you got, right? That is a specific theory known as alternative splicing mechanism. I'm splicing it alternatively. Okay, so that is how the one gene, one hypothesis theory was completely neglected and it has been rejected. So what is the significance of this? That there could be multiple proteins from the same gene and one protein can affect more than one phenotype. Okay, it is not that it will only affect one phenotype. One protein only can affect more than another phenotype. More than one phenotype means here one phenotype, other than that it may affect another phenotype. So the best way to describe it is when you have some diseases like, you can say, point mutation diseases. Point mutation diseases. So I'll go in little depth so that your school is going slow. So next class also we'll take genetics only. So let's go into little bit conceptual. Point mutation disease means what? Suppose there are two types of mutations. First of all, one is Mendelian, or two types of diseases, Mendelian diseases. Next you have chromosomal diseases. Two types of genetic diseases you have. Here you have point mutations. Okay, but number of chromosomes they remain constant. They remain constant forever. Okay, but here number of chromosome changes, no point mutation. Just the opposite of that. No mutation. Now, if you talk about point mutation diseases, means that you have a specific nucleotide or a specific base being mutated. Okay, like you have, suppose you have this gene. So here instead of A, I put suppose this one. So here instead of A, I have put C. So it is a point mutation. And later on, when I will be teaching you genetics codon chapter, okay, codons in genes, so we will be knowing that three group of bases or nucleotides they code for one amino acid. So suppose this is coding for an amino acid known as 16. Since this has been mutated now, this may code for alanine. Okay, just a or it is just an assumption. Okay, it may not code. Okay, so I'm just taking an assumption. You don't have to mug up which codon codes for what. Just an assumption. Suppose it coded for 16. This is coding for alanine because of this mutation. So what are you expecting? This is amino acid number 1, this is number 2, this is number 3, this is number 4. Suppose it has 100 amino acids in that specific polypeptide chain, and second amino acid is different. That means it's a missense mutation. And due to one amino acid, here first, here second, this one is the third, this one is the fourth, right? Due to difference in the amino acid, what happens is the protein configuration or the conformational stage or the quaternary structure of the protein, because if I show you any protein's quaternary structure, it is like this. So every protein works in its quaternary structure. Okay, just look at this image. So quaternary structure of protein. It looks like this. Okay, you can see this is how they look. Here you will be having active site, binding site. So if this is somewhat distorted because of one or two amino acid changes, then the protein, or suppose that protein is an enzyme or a hormone, that will become completely ineffective, completely useless. Okay, it may lose its function. Okay, loss of function mutation it is called. You have gain of, you can say, function mutations also, but that is way beyond your course. So this is loss of function mutation that I'm talking about. So it will not be able to do its job. It will not be able to perform its designated duty in the biomechanical or biochemical pathway. So thereby you will be having some problem or some specific ailments emanating in your human body, right? In that specific individual. So pleiotropy means, suppose there is a, I'll give you a very, very specific example for that. There is a disease known as phenylketonuria. Okay. Phenylketonuria. There is a disease known as one of the best examples of pleiotropy. What happens is there is an amino acid phenylalanine. It undergoes hydroxylation reaction, means addition of water and that OH, H2O. This is the H2O. So you will have that OH coming out, H will be out here, and OH will get added to this phenylalanine. Okay, it is known as hydroxylation. So there will be an enzyme known as phenylalanine hydroxylase that will convert phenylalanine to another amino acid tyrosine. Phenylalanine, tyrosine, okay, and tryptophan. These are the aromatic amino acids, means they have benzene ring. Okay. Now, in this gene, suppose the gene coding for phenylalanine hydroxylase has some specific point mutation, and thus hydroxylase enzyme has become completely useless. So this reaction cannot happen. You will have too much accumulation of phenylalanine. Okay. That will lead to urinary defect, mental defect, and all. That is one effect of phenylalanine. Another effect would be this tyrosine is the precursor of melanin. Okay, which is the skin pigmentation. You can say skin pigment contained by melanocytes. When tyrosine is not there, melanin will not be produced. Okay, and you will go towards more blondness. That is again another effect. One effect is mental retardation, another effect is you are going towards less synthesis of melanin, that is blondness. So one gene mutation, a mutation in one of the gene, missense mutation in one gene is causing two different phenotypes or not. Here, one is you are observing mental retardation, another way you are observing less melanin synthesis. Okay, so that is the meaning of pleiotropy. Okay, one gene's problem, a problem in one gene will lead to a myriad of phenotypic effects. Okay, that is known as pleiotropy. Okay, so this disease can cause mental retardation and reduced hair and skin pigmentation both. Okay, so this is one of the best ways to describe your pleiotropy. Any doubt, Isigar, in pleiotropy? Is pleiotropy completely clear? That how one gene can affect more than one phenotype. Okay, the missense mutation in one gene. That is the meaning of pleiotropy. So next we'll come on to, you can say, the polygenic inheritance. By the name only you can deduce what does it mean. Polygenic inheritance. How many genes? More than one gene. So if one character here, just opposite of pleiotropy, if one character slash phenotype is deduced by or it is influenced by more than one gene, or the products of more than one gene means more than one protein. If a culmination of proteins decide one trait, that is known as polygenic inheritance. Okay, so if you want to define it, you can write that when one phenotypic character is controlled by more than one gene, then it is called as polygenic inheritance. It is referred to as polygenic inheritance. Okay. So there is a scientist known as Colerentia. So Colerentia is known as the father of polygenic inheritance. It is also known by the name as quantitative inheritance. Okay. Quantitative inheritance. Because you can quantify, right? How many dominant alleles are present, okay, how the phenotype is being affected by the presence of various dominant alleles, okay. The dominant alleles will have cumulative effect, you can say, which will help in expressing one part of the trait. Okay, more dominant allele, the better will be the trait expressed. It will be having a cumulative effect. That is the application part of genetics towards expressing that trait, or part of the trait. Okay. So the genes involved in quantitative inheritance or polygenic inheritance are known as polygenes. Okay. So means multiple genes are causing expression of one phenotype. So those genes will be known as polygenes. And skin color of man, it is not decided by one gene only, multiple genes. Then wheat kernel color. So all these are the main paradigms or exemplars of polygenic inheritance. Okay, and it does change the Mendelian ratio also. Okay, so I'll just give you a perspective because the current entire derivation of the via cross is not in your course. Okay, so when two polygenes are considered for one character, okay, means two genes are considered simply, technically polygenes for one character or trait or phenotype slash phenotype, what happens is you have the ratio, okay, which is very, very different. Okay, the ratio is not something that you might have seen. It is 1 is to 4 is to 6 is to 4 is to 1. Okay, so that will be the ratio in case of polygenic inheritance. So this is the ratio that you get. Okay, so when there are three genes, the ratio will be even more. Okay, for three genes, I'm just writing down the ratio. No need to go for the derivation via the cross. Okay, 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1. So this is how genetics works in the higher classes. Okay, so that is how the genes work. Okay, one example, I'll give you one very simple example of this. Okay, not making it complicated. Suppose you might have heard of Negro. Negroes are very, very dark. They'll have high melanin concentration, right? So when you have a Negro suppose has two genes. A person who is Negro has two genes which decide the skin color. Suppose it is completely homozygous dominant for both. Okay, A and B both decide melanin, so they have high melanin. Okay, and I can refer to them as they are coming from the Negro descendancy. Okay, and suppose that girl, she marries